Okay, so welcome back everybody. This is the point at which we branch out into some other diseases of concern. So moving away from rusts and mildew. And our first presentation is in barley yellow dwarf virus, and this is to be given by Lawrence Bramham from Rothamsted Research. Just a little bit about Lawrence. He's the lead of the wheat insect virus interactions subgroup at Rothamsted Research, working within the BBSRC funded 5-year project Delivering Sustainable Wheat, which actually runs until 2028, as well as the DEFRA funded WIGIN project, which has been going for many years actually.
Yeah,
I've lost touch with how many years that's been on. So Lawrence is an experienced plant virologist, entomologist, and he's developed quite a lot of the techniques required for studying this and, for instance, disease diagnostic assays. So over to Lawrence.
Thank you very much, John.
Where's the laser pointer? There you go. So this is actually my first time attending one of these UKCPVS events, so it's fantastic to be given the opportunity to actually speak. I see... Hang on.
Just make sure
there we
you
go.
point that direction.
In that direction, brilliant, okay. Because I see these opportunities as such a fantastic way of actually networking within the room, because we've got so many expertise, and actually connecting the dots and seeing where actually all of our interrelated skills can actually come together to resolve the challenges that we face in terms of cereal production are really valuable. So this is a great opportunity. And speaking of the challenges that we actually face, so I'm here to discuss barley yellow dwarf virus. But it'd be slightly remiss of me not to discuss this topic without also discussing what's always lurking in the background, which is the aphids as the vector of the virus, of which there's one. So today I'm going to give a generally holistic overview of where we are in terms of barley yellow dwarf virus, the challenges that we face, and also the opportunities. And I'm hoping that throughout all of this, whether you're from a breeding background or an agronomic background, there'll be some useful nuggets in here for you to take home. I see collaborations as being the kind of bedrock of everything that we can do as far as getting real research out there. We've already mentioned WIGIN here, which I'm now involved in the 5th iteration. There's a series of 5 others, and this is mostly also just to highlight where I actually come from, for those of you who don't know me. So in my PhD, I started, first of all, in VEGIN, and then moved on to ORIGIN, very much working on the brassica side of things, where I really gained a strong understanding of actually it might be well and good having sources of genetic resistance, but unless you know what else is out there in terms of the viral threat, how do you make sure that they actually are retained as useful and going to last? And that's something which I've then brought in to the WIGIN side of things, where in its form iteration, WIGIN 4, BYDV was the focus, but now going forward until at least 2028, we're looking at the other viral threats that actually exist in the UK as well. And in this iteration, we've got a diverse management team, additional members in Mess Office who are doing a lot of kind of aphid modelling, trying to identify, as I say, what's really coming into the UK and is going to be potentially future targets. This is a collaboration network that you might not be aware of. This is more of an informal one of which I'm a member. This is the UK Yellow Dwarf Virus Network. I won't go through the full list, but the idea of this is to really bring expertise together on BYDV and other yellow dwarf viruses, mostly in wheat, but other cereals, and try and identify where our resources can actually intermingle and cross-connect. Because a lot of the time we're asking and trying to answer the same questions from different pathosystems and different crop species. There's already been some good outputs coming from this. For example, this, which is the first output, looking at a modelling framework. This is in BYDV. I think these will be shared after, so you can have a look at that. It's very much a modelling paper, but it touches on a lot of the things that I'm also going to expand on today, thinking about viruses and the aphids and the wheat genetics as an interconnected system, and that really to tackle this threat, we need to look at it holistically rather than just looking at one facet at a time. Another thing that I'm involved in is the epidemic preparedness team. It's this, I was literally just there last Thursday and Friday, so I haven't been able to add much into this set of slides around this. But this is a team brought together, Thursday and Friday rather. We had 20 expertise tasked with answering this question. It was one of 11 funded from UKRI, looking at interdisciplinary research proposals to tackle epidemic threats. All of the others are based on animal and microbial systems, so we're the only one that's looking from the plant side of things, and I think there'll be outputs from this that can be, again, directly applied to a cereal and wheat system. And then finally, but by no means least, I already mentioned the wheat insect virus interactions, so close to home. This is formed of myself and Isiana, who I'm very pleased to say is also in the room with us today. And together, we work in the wheat aphid virus interactions pathosystem. But for the purposes of today, I'm going to mostly focus on the diagnostics and the virus side of things, but I'll mention some of Isiana's amazing work as well. So, talked a bit around it, but Barley yellow dwarf virus, to bring you all up to speed in terms of this pathogen, it is a particularly insidious pathogen for a number of reasons. It causes a quite high yield loss, averaging about 30% in field conditions, but this can vary on a huge number of factors, whether it's the strain that's infecting, whether you're getting co-strain infection of a susceptible genotype, and also the age at which the plant is exposed to the viral pathogen. It is quite challenging to actually identify in the field as well, and for that purposes I've got a prop of some wheat leaves that were infected with the virus. If you want to come to me in the break and have a look at those, feel free. But a challenge that often happens when I speak with agronomists, they often mention, well, I'm seeing symptoms that might be BYDV, might not. How do I really know what I'm seeing? And without proper diagnostics, it's tricky. Normally I'd say at this stage it's just a general challenge, but to try and get a bit of something out of this, I thought I'd just highlight a few tips for you once you get back out into the field. So this isn't a golden way of doing it, but if you answer 2 quick questions of looking at the tissue, are you seeing symptoms that's a bit stronger rather than in the plant tissue right at the base? If so, it's not as likely to be due to senescence. And the other thing that I see that's characteristic of BYDV, whether it's yellowing or that beautiful kind of purplish discoloration, is are you seeing discoloration going down the leaf in a V shape? And if you see both of those things, then I would suggest it is likely to be BYDV. And at that stage, please do sample and feel free to send it to myself and I'll test and get you some detailed information back. Back to BYDV though. In terms of control, there is no practical virus-focused chemical control. There are some really cool techniques and technologies that are up and coming, like RNAi-based approaches, but unlike some pathogens like fungi, where you can apply a fungicide, we have nothing that can directly target the virus itself at scale, at the moment anyway. And for that reason, the aphid vectors, so what are actually spreading the virus, are instead targeted, as well as developing resistant cultivars. And the reason I've got here resistant in inverted commas is because there is some debate on what is actually really true resistance, what is tolerance, and what does that mean as far as BYDV control. I'll go on to discuss that shortly as well. But something that is often forgotten about, and which I've already tried to kind of allude to already, is that the interactions between the aphid vector species that are spreading the virus and different BYDV strains are incredibly complex. And it's an area of work that we really need to progress forwards with if we're really going to tackle this significant threat. And a reason for this is the way that actually the viruses are spread. So simply put, you get some viruses that are done in a non-persistent manner. This is kind of the easy version where the virus just goes, adheres to the feeding mouthparts, is very quickly adhered, very quickly lost. This has challenges in terms of actually insecticide control. You need to have an insecticide that can kill the aphid quickly, but the aphids lose the virus over time. That's one situation. BYDV isn't this. BYDV is instead persistent. And what this means is that there's a period of time where the aphid isn't necessarily viruliferous or able to pass on the infection. It uptakes the aphid into its hindgut. It then translocates into the hemolymph and the salivary glands. And after that point, the virus is able to be spread. This does mean that the aphid carries it for life, which has challenges in itself, especially in terms of overwintering of populations. We're facing situations now, much as it might be nice and sunny or relatively cold outside, overwintering is going to become more and more of a threat. We're going to have strains from one year going to the next, and for something like BYDV, where the aphid, as I say, keeps it forever, we need to keep an eye on what's actually out there. Another factor as well is that not all aphids are the same. Different aphid species will vector different BYDV strains, either qualitatively able to or not, or with varying efficiency. And even within certain species of viruses, you'll get different efficiencies based on populations. So as we're changing the population structure of aphids across the UK, this is something we need to keep an eye on. Which again leads on to the challenges. And we've got some data here from the most recent Met Office looking at the mean minimum temperature for 2023 across the difference between 1991 to 2020. And generally speaking, you can see most of this is in pink. This is posing challenges as far as overwintering is concerned, and climate change, as we all know, is the big elephant in the room as far as what's going to change in the future. And with this being a pathogen that is spread by insects, it's something that really needs to be considered heavily. As far as the strains goes, there are a series of strains of BYDV. These are the major ones that have been historically prevalent in Europe. In the UK, it's mostly been PAV and MAV as the core strains of interest, mostly because these are spread very efficiently by the two major aphid species in the UK, for BYDV at least, which are Rothamsted paddy and Cytobia novini. But then there's also been these ones kind of lurking in the background, and no one's really been properly assessing what is in the UK, where and when. And that's kind of where my work has been coming in. If you take BYDV, MAV and PAV and look on a bit more of a global scale, what's going on, not all the viruses are created equally. There are some evidences of different virulences associated. And this is really just to highlight that if you take data from resources like NCBI, you get variation. This is challenging in terms of actually developing better diagnostics, and especially how do you resolve false negatives. So if you develop a diagnostic based on, let's say, the data for here, and then actually it's these sort of variants that are present in the UK, you don't detect it. Is it that the virus isn't there, or are your diagnostics just not fit for purpose? And that's something that I really wanted to get resolved with the work that we've been pursuing. Touched on it already, climate change, anything and everything that we don't want to be happening to aphids will be happening in terms of it being detrimental. The major impact is going to be temperature, increase of things like aphid forms, so they kind of move away from the wingless forms that the alates, higher virus spread, more movement, dropping rates from plants, so them actually coming off the plants and moving. But one of the major things is generation time. We'll have more generations occurring, higher numbers of aphids, so we'll just have generally more aphids around. This doesn't include what I said earlier, though, about the overwintering aspect, and that, from the BYDV side of things, is perhaps going to be the most influential. But all of which creates this perfect storm where we really do need to have better BYDV management and a better knowledge of what we have in the UK and what's going to be in the UK in future. Looking historically, I love a chart that you can interpret in less than a second. That's a lot of blue. This is from the UK pesticide usage survey reports for the last 10 years. Aphids are the target for foliar insecticide application, at least in wheat. We are changing the population structure of aphids over time with the selection pressure of insecticides. How is this going to change BYDV and its ability to be spread when we're actually narrowing down the genetic pool of aphids potentially? Another alarming thing as far as BYDV control is everything in blue here is pyrethroid-based active ingredients. And this is based again on the most recent UK pesticide usage survey reports. We have a limited arsenal available, and that's going to reduce over time. If you look at the two aphid species that I mentioned earlier, we have already moderate pyrethroid resistance widespread across Cytobia novini, and there's already been evidence of pyrethroid tolerance in our paddy, and these are going back a fair bit now. We need more testing for resistance, as well as also testing for general things like the viruses, to see how our tools are going to be useful in the future. But that sounds all a bit doom and gloom. We do have resources available to us. I'd say the first and foremost of which is BYDV resistance, so genetic sources of resistance in the plants themselves to resist the viruses. But there are nuances to this that need to be resolved. So, for example, if we've got a source of resistance, do we know what BYDV strains are out there, their distribution? What is the specific mode of action? Are they going to be resistant to all of the spectra of the viruses? And a component here that is very rarely considered is actually co-infection. So you develop a source of amazing resistance to BYDV strain X, but then X and Y come in. Does it overcome it? We had a summer student a couple of years back that was looking at co-infection of different genotypes of plants with different strains of the virus, and we did notice trends that if you introduced one and then a few weeks later another, it potentially overcame resistance, or if nothing else, influenced the virulence of severity of the disease going forwards. So we don't need to just consider individual strains. Actually, the mixing of strains is also of extreme relevance. Now, resistance or tolerance is up for debate, often I'm going to break this down very simply. So you can split it based on the response of our plants to viral challenge, infectable versus non-infectable. That's nice and easy. Behaviour of the virus, so susceptible versus resistant. But where things get a little bit more nuanced is when you actually look at the plant disease response. And here you can have a kind of mess of a situation where you have a plant that is susceptible to the virus and extremely sensitive, so you get huge symptoms emerging. But you can also have sources of resistance, which are resistant to one strain of the virus, but then when another strain comes along, the plant hyper-responds and you get extreme sensitivity, that plant dies. Now, in one terms of controlling the disease, that could be a good thing. You kill that plant off, the source of inoculum is gone, it can't be spread, but that'd be a hard argument to be made to a grower to say, oh, grow it in your field, if it comes down with the virus, you'll lose that bit of the crop, but the rest is okay. It's tricky. On the other side of things, tolerance, you can have susceptible varieties that have got no actual genetic source of resistance, but there's little or no effect. And likewise, colloquially resistance, and again, tolerance as well. As touched on earlier, the fact of planting dates being important. A lot of varieties of plants, even though they're infectable and susceptible, they might be tolerant after a certain growth stage, such as about growth stage 31. But there's challenges in terms of, well, do you grow early, try and bypass the aphid population moving in? There's lots of nuances to it, and it's something that we really need to explore further. So where are we in terms of BYDV resistance? There are a fair few, but they're relatively scarce. So in barley, for example, there's 4 genes that are linked to tolerance or resistance, named RYD1 through 4B, and a few QTLs as well. RYD2 is the most heavily deployed, and I'd argue works quite effectively. It reduces the viral titer in young plants. There's some doubt from testing of older plants, but the virus can spread systemically still. So those individual plants, whilst they are not coming down with the disease, they're going to act as an inoculum going forwards. Might that influence the BYDV that's available? Might it drive evolution for new resistance-breaking varieties? It's all aspects that we need to properly explore. Meanwhile, in wheat, similarly, there's 4 BYDV resistance genes, BDV1 rather, through to BDV4. BDV2 originated from this grass ancestral species, and through the efforts of RAGT, the first winter wheat commercial variety with BYDV resistance was deployed in what is now branded Gencerus. This is the one that, as a group, we know most about, because Isiana is doing some excellent work tracking down this source of resistance and exploring its resistance mode of action. The way that this is being pursued is a really innovative approach. Rather than doing QTL mapping and having population development and refining on the gene, she's taking forward the source of resistance, mutagenizing it to interrupt various genes across the genome, and in some instances, identifying susceptibility. And this is where the mode of action has been effectively interrupted. And so now we're at a stage where we're doing the sequencing and we'll be comparing BYDV resistant and susceptible versions of the original Wolverine resistance to really narrow down exactly where that resistance is and hopefully allow it to be more readily deployed. But this all comes back to that major question of what is out there, both in terms of the aphids and also the viral strains. We're very lucky at Rothamsted to have the amazing resource, which is the Rothamsted Insect Survey. So for those of you who aren't aware of this, this is a network of 16 suction traps, which are these 12-metre tall, effectively hoovers, constantly sucking in any aphids or small insects that fly overhead. There's an amazing team who take these every week, separate the bycatch, so all the stuff they're not interested in, and then through microscopes, analyse, assess, and categorise what they're actually collecting on a weekly basis. This is an amazing resource in terms of decision support tools, but it's also fantastic for scientists like myself to take those samples forward and see what's actually in the aphids themselves. It generates amazing data like this, where we can actually look at the different aphid pressure over time. So this is for 2023. I'll have 2024 on the next slide. But you can see even this relatively basic overview of what's collected across the entire network, different trends emerging. So, for example, Satobi and Avini peaking around June, July time, whereas paddy peaking more in the autumn time. And this correlates with whatever's going on in terms of the field and disturbing the aphids, harvesting, etc. But is that point of, even if we just do it in terms of simple, that aphid is carrying that virus strain, that aphid is carrying this, we do need to think of things more on a dynamic, geographical, temporal basis, as well as what genotypes are occurring in the field. It's a complicated system, but one which we have got the tools now to properly explore. This is for 2024, so you can see already if you compare 2023 to 2024, there's a shift in peak there, whereas there's less of a peak for paddy. But it's all of these inter-year comparisons that need to be fed into other amazing tools like decision support systems so that we have real-time monitoring and real-time ability to react to the aphid and viral pressures. This is some historic data collected by my colleague Martin Williamson, who is actually very recently retired from Rothamsted, along with Stephen Foster, for those of you who know Stephen. So it's a bit up in the air as to how we're going to be pursuing this going forwards. But as I say, we've got some amazing resources and tools available, so I'm hoping that we can pursue something quite exciting, or even if we don't pursue something exactly like this, something different in future. But this gives a holistic overview of actually, we're detecting BYDV UK-wide. Roughly a fifth of all aphids that are captured are infected with BYDV. This isn't looking at the individual strains, this is just qualitative presence-absence, but it does vary. So, for example, we have low instance, 5% was the lowest, that was back in 2021. Meanwhile, meanwhile, highest instance, virtually to the date a year before, 46% in the same aphid species. So we do need a dynamic monitoring system. What I've been doing, though, is taking a subset of these aphids forward, doing Barley dwarf virus sequencing, specifically of the coprotein region, to try and get better diagnostics and an idea of what, I say, is out there. And this is kind of where we're at the moment. So all of the aphids that I test are from the autumn, so we're still processing the 2024 ones, but this gives you a bit of a kind of insight into some trends that are emerging. There are caveats to this data. For example, I wouldn't consider this representative truly of what is out there as a comprehensive sampling process. This was more just to see the variants that might be emerging. But yeah, some interesting things. So first and foremost is, I've mentioned previously BYDV MAV and PAV, the two strains. Suddenly, we've got this green one, BYDV PAS emerging. Prior to doing this work, PAS wasn't recognised as in the UK whatsoever. It was present in Europe, so at some point there has been an influx of this variant. There's a question mark over whether the current ELISAs and other serological molecular diagnostics are potentially picking this up. The good news is I think most of the time they are. This PAS is very similar to PAV, both in virulence and resistance spectral status, but it's something we need to keep an eye on. Another thing to note is that there's certain areas like, for example, Hereford, where the distribution of these strains is it's relatively conserved. There's not that much difference year on year. Meanwhile, there's other areas like York where something's going on. So we started with a mix. In 2021, it seemed to be near 100% MAV. 2022, of all the 12 aphids, both Stobionavini and our paddy that I collected, were all carrying BYDV MAV. And then most recently in 2023, yes, a bit of PAV and PAS coming in, but it's mostly MAV. This has huge implications for resistance screening and germplasm development and deployment, as well as also looking at just aphid populations in general. Why is it that we're seeing a trend going on there? And then similarly in places like Broom's Barn, where there's a bit of a mix and it's dominating the other way. This is all stuff that we need to properly explore in order to really get a handle on what's going on. You might remember this slide from earlier, but this is just to highlight that with our work, we've identified PAS both in our paddy and Stobionavini, as well as validated the presence of MAV, PAV, GAV in Stobionavini, and the standard Variola dwarf virus in our paddy. But where I think this sort of data is really coming into its own is the diagnostic development, and that's where I'm really prioritising my efforts before hopefully deploying it on a higher throughput spectrum setup in the future. So I would state this data is anonymised for various reasons, but it is representative of what we're seeing, and this is just a subset of the 2020-2021 data, just to highlight that we're seeing quite a lot of uniformity in MAV, a little bit of variation in PAV, but the drive-home message of this is we have identified conserved UK strain-associated polymorphisms that are linked with BYDV, MAV, PAS, and PAV. And that's allowed me to develop a whole suite of diagnostics, such as this one, which is the TACMAN-based one, incredibly valuable and robust diagnostic for in the lab. But alongside this as well, CASP genotyping chemistry for if those in industry wanted to deploy this. So this is more of a one that's used in genotyping, as well as routine PCR and qPCR assays for each of the individual strains. Very recently, we were awarded a BSPP small project fund, and the purpose of this is trying to get this resource out of the lab and actually into the field through the use of isothermal, so single temperature diagnostics like RT-LAMP and a CRISPR-Cas13a based assay, reaching a stage where potentially within about half an hour, you could take an aphid or a leaf, put it in a one tube reaction, put it into a little box that's got a source of UV fluorescence and get a quick indication of that plant or aphid has BYDV in it or not. And that's where we're wanting to really get to. As far as actually rolling out a higher level of throughput diagnostics, we're in a good place. We've recently got this new piece of kit, and the way that this works is that you can actually process, rather than single well, single qPCR assay, this will allow you through the amazing magic almost of microfluidics for every sample to be tested against every assay that you put in it. So the most useful version of this would be up to 24 separate molecular assays looking at insecticide resistance, strain 1, 2, 3 of BYDV, other viruses that might be spread by the aphids, against up to 192 samples. And this would generate qPCR data in 2 hours, 4,608 qPCRs on a single plate. And this is where I'm really wanting to take this basic work forwards. So in terms of actually bringing this back, and this is my final slide, to yourselves in the room, and potentially sampling for better BYDV detection as well as what's going on, I've been looking at aphids. I've not really been looking at actually what's going on in the field, in the plants. So I'm missing a major component. A lot of people often reach out to me saying, oh, can I just send you a quick sample? Yes, please do. But this is just to highlight a very simple experiment we did to see where you could sample, where you couldn't, and it came up with some quite surprising results. So we took a plant, specifically cultivar solstice, due to its rapid development, challenged it with BYDV, maintained it for quite a while until the flag leaf developed, and then took samples of both the challenged leaf and the flag leaf. And the idea of this was really, can you sample both? Will you get BYDV out of both? Are there some points in the tissue that are better to test than others? We get it relatively standardized, just taking equal amounts of tissue going in, but apart from that, keeping quantification and other quality control the same. And we were really genuinely quite surprised at what came out. So these are the qPCR CT values, so lower equals higher viral titer. There is a crude RNA extraction at the bottom, which was very much just mushing the plant tissue up in a buffer and taking it forward for the reaction. Surprisingly, this came out as actually you could do reasonably decent qualitative detection. These are all such high qPCR values, I wouldn't truly use it as an indication of titer, but we could get BYDV out and confirm the presence, even in tissue like this, which was borderline necrotic. But the thing that I found most interesting was actually on the more higher quality RNA extractions, they were virtually the same between the flag leaf and the challenged tissue. So the drive home point from this is, if you come across something in the field that's potentially got BYDV, do not worry too much about where you're actually sampling. Pop a leaf in the post, get it to me, and I should be able to get BYDV out of it and confirm presence, absence, and potentially what strains are available. So I think that brings me up to my time. So as conclusions, there are a lot of challenges that exist, both now and in the future. Shifting BYDV strains, different aphid populations, climate change is a massive one, as well as actually ensuring that the resistances or tolerances that we have are deployed effectively and last. But we're in a situation where we actually have a lot of opportunities as well. We've got the resources rather for modelling changes to this important path system and a lot of new tools. Something that I've not had time to mention today, but Isiana is pursuing the development of BYDV infectious clones for the UK as well. So those are an incredibly valuable tool. And we've also got the high throughput molecular assays. But as I say, I would gratefully receive any BYDV positive samples that you may come across in the field. I'd be happy to feed you back some valuable information as well. So thank you very much, both for your attention today and the UKCPVS stakeholder event organisers. Too many other people to thank, but thanks especially to Kim Hwan-Cossack, the leader of the wheat pathogenomics team, of which I lead the subgroup, as well as Isiana, who's here today. And please do go and have a chat with Isana about her amazing work, as well as continued assistance from the wider wheat and cereals community. So thank you all very much.
Okay, so we're moving on now to this title. I like this title, about a medieval mycotoxin. Very good. Joe Brennan is going to introduce the topic of ergot, and we've got two other ergot talks following on. So just to introduce Joe, make sure I've got my notes here. Joe is actually the head of technical and regulatory affairs at UK Flour Millers. He leads the team covering food safety, grain assurance, wheat variety development, sustainability and trade policy, acting as the primary liaison between the association and relevant government and supply chain stakeholders, pre and post mill. And he currently chairs the UK Ergot Working Group. So welcome, Joe.
Thank you, John. Good afternoon, everyone. That's very much the introduction I was planning on giving for myself. So I think the key thing is to say that I'm not a farmer, I'm not an agronomist, but I do spend a lot of time translating or trying to translate what goes on farms to the customers of our members. Now that might sound like an easy task. I mean, people know what farming is, you think, but actually people don't really understand it at all, and they set requirements and expectations that simply cannot be met on a conventional farming system. So today I'm going to go through, I think, why ergot matters to cereal processors. For those of you who are farmers or merchants in the room, I'm sure you've had a rejection or two. There's no glass screen in front of me today, so I'm hoping there'll be no missiles launched my way. I've had to speak to farmers before about ergots, increasingly in recent years, because as we all know, it's becoming more and more common. But I'm hoping today I can sort of demystify a bit as to why it matters to processors, why the strict requirements at intake exist. So to give you, that's the intro slide again. To give you a very quick overview of UK Flour Millers, we're the trade association for the UK milling industry. We've got about 32 members, 51 flour mills, and about 99% of production by volume. So I'm presenting a processor perspective, but it's really just from the flour milling industry, and it's also informed by my work on the Ergot Working Group. But that's not to say that the millers are unique in this respect. Indeed, if you supply malting barley, or even if you supply some animal feed homes nowadays, you'll also know the very strict tolerances for ergot at intakes there too. Millers in the UK, 5 million tonnes of wheat, of which in a typical year about 4 million tonnes is homegrown, a bit less over the past 2 years. And wheat flour is very much a staple food in the UK. It's in about a third of food products sold in the supermarkets, and it accounts for about 20% of the nation's food energy intake. So ergot, a very brief introduction, and I've got a sort of glossary of terms up here because people use the term ergot interchangeably in different contexts to mean related but different things. Ergot itself is the fungal disease that affects cereals. Ergot sclerotia are the sort of purple-black masses of fungi that the disease will produce. Ergot alkaloids are the mycotoxins that you find within those sclerotia, and that's what we're most interested in as processors. And then ergotism is the disease you get if you consume too many ergot alkaloids. Now, ergotism has quite an interesting history as a disease. One of the reasons for that is because ergot causes, if you have too much ergot, you hallucinate, essentially. One of the alkaloids is a precursor to LSD, or acid, as it may be known. And essentially, it causes people to see things that are not there. I wouldn't recommend, if you find yourself on a Friday night and you forgot to get booze at the shops, I wouldn't recommend tucking into some ergot, because before you hallucinate, it will cause quite severe vasoconstriction in your extremities, so you lose circulation to your fingers and toes, and you get gangrene, and it all sounds very unpleasant. We have got a big box of it at our office, and there's occasions where you're tempted and you think, oh no, maybe better not. But you know, that because it causes those hallucinations, it has a very interesting, very storied history as a disease. The middle image is a dancing plague, I think from Belgium or France, which was these incidences where whole villages would just start dancing until they died of exhaustion. And ergot has been linked potentially to what was causing that with the way it causes involuntary spasms and all these things. It's also been linked to witch trials. You know, you can imagine if a village has eaten some contaminated grain with ergot, they start seeing things, the livestock start dying off, they're going to blame a witch. Unfortunately, that one seems it doesn't seem to hold water when modern historians have a proper look at it, which is a shame, but it's a nice little story. And then there was a flour mill in Oxfordshire about 10 years ago, and they were contacted by essentially a pagan cult. They found out that this mill had ergot sclerotia on the site, and they wanted to buy it for a werewolf transformation ritual. And that sounds made up, but it is actually true. It was a werewolf transformation ritual. So they didn't sell them the ergot, although they probably get more money than you get for flour. So it has a very sort of storied, quite a theatrical, sinister history as a disease, but many of you will not know anything about ergot. So why is it relevant in the modern context? So one of the reasons is that ergot as a disease is increasingly prevalent on farms in the UK and across Europe. That has unfortunately coincided with the establishment of maximum limits for ergot alkaloids in processed cereal products, affecting the EU and Northern Ireland. And of course, because of the increased prevalence, because of the controls in the supply chain, there's a massively increased cost in terms of cleaning and also rejections when grain containing ergot is delivered to a home that can't accept it. So this is a very brief and fairly boring slide about the ergot and ergot alkaloid limits. They came into effect on the 1st of January 2022. That was just after the UK had left the EU, so these were not carried into GB law. But they do apply to Northern Ireland and to EU member states. So that's to say that there are now ergot alkaloid limits akin to sort of DON or ZON limits in processed cereal products, so flour, oat-based products, any cereal except maize and rye. These limits exist, and they exist in Northern Ireland and the EU. So although they're not applying directly in GB, many customers will set their specifications in line with what's going on in the EU. And that's because it's very difficult for many customers in the UK to segregate their flour or whatever product they're using. So essentially, they treat the markets as one. So these are having a massive effect in the UK market, affecting an enormous proportion of the cereal products that millers and others will produce. So the challenge with ergot is that it's very difficult to control the alkaloids at the limits at and around the concentrations of the limits. The limit in white flour is 100 parts per billion, which may sound like a lot, but it's not at all when you consider that research found the average sclerotia contains 1.9 million parts per billion ergot alkaloids. So if you even get a small piece of that sclerotia into the sort of grist that you're milling into the flour, you can see a massive spike of alkaloids and your flour will be non-compliant. And this is a food safety non-compliance. There have been product recalls in France, in Belgium, in many European countries because of alkaloids in processed cereal products. The challenge here is really that when the European Commission set these limits, I don't think they quite understood what they were looking at when it came to the relevant levels of sclerotia in grain. Essentially, there's a mismatch between the sclerotia limit, which is now 0.02%. That's actually after cleaning, so a processor is entitled, if they want to clean grain, and if it's got less than 0.0, it can have up to 0.02%, and they can still mill it into a product. But you would never do that. So the table on the right-hand side shows you what, based on the observations of the average level of alkaloids in a wheat sclerotia, what the ergot alkaloid concentration across that bulk would be. So if a mill was to choose to mill at 0.02% grain containing that level of sclerotia, they could end up with a flour at around 377 parts per billion, which is well over the 100 part per billion limit. That 100 part per billion limit, by the way, is dropping to 50 from 2028. So the limit's already really difficult to comply with, and it's being halved. We did some work where we spiked grain that was completely clean of ergot with 0.02% sclerotia. We milled it into a white flour, and then we tested that white flour to see what level of alkaloids was in there. And we found it was around 350, just under 350 ppb on average. So again, well over the legal limit. So it's quite difficult to really see what they were thinking when they allowed a tolerance in grain that would guarantee a non-compliance in the grain-based product. We said to the European Commission at the time, and we've been working on this for about 10 years now, They probably hate us by now. We said to them at the time, this is a lovely graph. It's showing an excellent relationship, but actually the level at which ergot is relevant, the level at which the alkaloids are relevant, is an incredibly noisy patch in the bottom left of that graph, where you can see there is a greater deal of variability, and we don't think they accounted for that variability. So as I said, in the UK, I come to a lot of conferences and I get shouted at by farmers because they've sent grain to a mill and it's been rejected. And sometimes they've cleaned the grain and it's been rejected. And I can understand that frustration. From their perspective, they've done everything right. They've had the grain cleaned, but they're still seeing a rejection. So as I say, many mills have a zero tolerance for ergot sclerotia. The vast majority have a tolerance that's well below 0.02%. But despite that, we are seeing since 2021 very high average levels of ergot alkaloids in wheat. And I should say, this is wheat that's been accepted into flour mills. So this, for the vast majority of these samples, would have been wheat that had no visible sclerotia at the intake. And yet we're still seeing levels of alkaloids in there, which if you were to mill that into a flour, you could be at risk of exceeding the legal limit in the flour. So that is one of the reasons why mills are being very strict on sclerotia. I shouldn't say just mills. I mean, maltsters and some feed millers are the same as well. It's across the cereal processing sphere. But that really does pose a significant compliance challenge. It also highlights, you can see the wholemeal flour limit there, the white flour limit, and the one that will be reduced in just under 4 years' time. It shows you how strict those limits are relative to what we're finding in grain at the moment. And the graph also demonstrates, I think, the rise in ergot alkaloids that we're seeing. That's 3 years, or 3 of the past 4 years, we've got exceptionally high levels of alkaloids, and that's really challenging. So, despite the fact that we've got this process of being quite strict at intake with rejections, despite the fact that every load that arrives at the flour mill will be visually inspected using an agreed industry method for ergot, we're still finding very high levels. So what's explaining this? And part of it is that we don't still fully understand how the alkaloids are contaminating the grain. We know that the sclerotia contain very high levels of the alkaloids, and that's thanks to work that NIAB did in 2019. It was a 1-year research project. I think it was about 40 grand, AHDB funded, and that really massively expanded our understanding of how ergot alkaloids contaminate grain. But as is often the case, when you find out loads of things that are new, it sort of starts to beg a lot of new questions, and we haven't really built on that since then. But that research found that when you tumble or disturb a pile of grain with sclerotia pieces in there, those pieces, and then clean that pile of grain, you still get elevated levels of alkaloids in that grain, which suggests then that it could be effective friction, rubbing the sclerotia, or it could be the sclerotia fragmenting into smaller pieces, and you get tiny fragments of sclerotia that pump up the levels of alkaloids. We also, the researchers found that ergot infections above and below, grain above and below the point of infection in the ear contained a background level of ergot alkaloids. And that could vary quite significantly from, I think it was 25 ppb all the way up to 160,000 ppb. But it was a relatively small number of samples, and it really begs a fuller research project to look into that. Now, those are really challenging forms of ergot alkaloid contamination because they're essentially invisible to a mill intake. Actually, the easiest way to tackle sclerotia, or the dream scenario, if there is one, would be if all sclerotia were enormous and didn't break up, because they'd be really easy to clean out of the grain. But unfortunately, that isn't the case. A lot of sclerotia are small, they break apart very easily. And now, and I don't want to steal, Leslie, too much of your thunder, but now with blackgrass and the grass weeds that are very favoured hosts of ergot, we get very small pieces of sclerotia, very thin. They can break apart very easily, and they're really challenging in terms of the contamination or the transfer of those alkaloids into the grain. There is no rapid test available for ergot alkaloids at an intake. So unlike deoxynivalenol and zearalenone, you can't very easily at a mill intake test grain and determine that it's going to be compliant. So, in a way, we are relying almost entirely on detection of sclerotia at an intake. But then if grain cleaning is not delivering a reduction in alkaloids, or as significant a reduction as we would like to see, where does that leave a cereal processor? It becomes almost a case of sort of compliance Russian roulette. And I should say, going back to this graph, I'm slightly conscious of, oh, it doesn't allow you to go back. Oh, there we go. I thought it was an intentional thing. You know, that increase, by the way, since 2021, there's been a massive investment in grain cleaning by farmers, by merchants, and by cereal processors. And despite that, we're still seeing these enormous levels of alkaloids. So that is another aspect to this challenge, is why is grain cleaning not as effective as we had hoped? Is it the case that because you're disturbing the grain as it's being cleaned, you are just increasing the fragmentation of sclerotia into those smaller pieces? Is it that with the new greater prevalence of grassweed ergot, colour sorters, the sort of gold standard for removing sclerotia, are not as effective because those grassweed ergots they're far less visually distinct and the coloration is less distinct. There are all these questions that are emerging, but it's a very worrying scenario for cereal processors fundamentally, and the fallback is to rely on as strict a tolerance for ergot at intake as possible. So what's the sort of next step really? Well, I think tackling ergot at source is going to be really important. You know, we've seen that cleaning is effective in reducing the ergot alkaloid contamination, but given how strict the limits are, cleaning is not necessarily delivering the control that is required. Part of that could be that, as we said, the infections of ergot in the field, in the ear, develop those sort of invisible alkaloids in grain that is visually completely healthy above and below that point of infection. When you're cleaning, you can fragment those ergot sclerotia pieces into smaller bits, which could be increasing the alkaloid loading of the wheat itself. But of course, you know, ergot is costing a lot of money. It's very difficult. Farmers are very annoyed. So why is it not being dealt with? Well, of course, the on-farm control for ergot is very challenging. There is no, to our knowledge, there's no spray that you can apply that will deal with ergot. And a lot of the management recommendations, there's a friction now where farmers are being encouraged either via policy or subsidy or customer requirement to farm in a more sustainable manner. So something like minimum tillage, no tillage, flowering grass margins. There is some evidence that, you know, some of the control measures for controlling ergot, deep cultivation, for example, to bury those sclerotia under the soil, there's a friction there with where farmers are being encouraged to go, and that's really challenging. So some interventions are not appropriate for all farms. The contribution of risk factors is, I think, relatively poorly understood in the UK. If you're a farmer and you're committed to an SFI payment because you don't want to cultivate or carry out deep cultivations, then you do it and it doesn't even solve your ergot problem. You know, that's a real kick in the teeth. As I said, the agrochemistry is very limited, and I understand we've spoken to agrochemical experts, and they're saying there's not really much coming through the pipeline for ergot, which is quite worrying. So it is a massive challenge, and further research is really needed here. You know, we've got 4 years until those EU limits halve. As I say, they're EU limits, but they apply to Northern Ireland and EU member states, and that reflects an enormous proportion of our demand for cereals and where our products go. So we really need some research ASAP. We're almost sort of, we're behind schedule almost, which is strange to say when you've got 4 years ahead of you. But the review of AHDB's guidance is really welcome. We as the Ergot Working Group pushed AHDB to do that, and we hope that not only will it update the guidance and give farmers the best current recommendations for tackling ergot, but it can also hopefully identify sort of where the knowledge gaps are and where farmers need to be going to next. So that's it for me. I'll just summarise very quickly with the key points. Ergot's a food safety issue. You know, we're not just rejecting for the sake of rejecting. That would be ridiculous. It costs mills a lot of money too. It's a food safety issue, unlike the other mycotoxins where the maximum levels in the likes of flour are relatively achievable, you know, barring exceptionally poor years. With ergot, we saw that they were set at a very, very strict level, and they're posing a significant compliance challenge even in a regular season. The prevalence of ergot in the UK is going up. France are having the same issues too, I understand. You know, they've just had their worst year for ergot, which you know, some would say that 2024 in the UK was our worst year as well. And that despite the fact that Grain cleaning, there's been a massive investment across the supply chain in grain cleaning. The alkaloid levels in grain that is being accepted into cereal processors is rising, which is very worrying. I think I've covered point 3. Point 4, grain can be contaminated with ergot alkaloids in different ways. We don't understand fully how that's happening, and we need some more research in that area to better understand it. I mean, I mentioned the NIAB project was only 1 year, 40 grand, and it just told us so much, which I think shows how ripe ergot is as an area for investment. It maybe hasn't received such a big focus because I don't think it has a massive yield impact. But now the cost of cleaning, the cost of rejections, that has to be factored in terms of prioritization of this disease, as well as the fact that there is a food safety implication too. And as I say, tackling ergot at source, preventing those infections is going to be key. Grain cleaning costs a lot of money, and it may not be optimal in terms of removing the alkaloids from the grain. So the more we can do to stop the infections happening in the first place, the better. So that's it. I'm very happy for my slides to be shared. My email's on there. Happy to take questions now, but if I can't answer it and you don't see me at lunch, just send me an email or give me a ring.
Okay, so let's move on to Leslie Boyd. Just quickly, I think Leslie is well known to many of us, but I should do some sort of introduction. She was a research group leader at the John Innes Centre with a remit to study cereal rust interactions and has been closely involved with the work we talked about earlier. In 2012, she moved her research programme to NIAB, and since 2014, she's assumed management of the cereal ergot research programme. So over to you, Leslie.
I must warn you, John, I had heard from a colleague at the UK Ergot Working Group that told me he found a sclerotia in his muesli.
Yeah.
So do check. Right, so presentation, please. Thank you. Okay. So, what is ergot? Challenges and solutions. Right. So I just want to start, as John kindly said, I took over this programme in 2014. It was started by Rosemary Bales and Donald O'Sullivan, but it was mainly led by, I've got a pointer, this lady here, Anna Gordon, who did most of the work, but she's also now left NIAB. So it's up to me to keep the programme going. But I want to thank many people that have been involved in a lot of the work that I will present this morning. So, what is ergot? Where does the primary and secondary sources of infection come from in the farmer's field? How do we manage ergot on farm? Can we look to ergot resistance, and why are we not breeding for resistance in the UK? I believe in Germany there are programmes that involve breeding for resistance. And ergot alkaloid synthesis and contamination of grain. As Joe said, how is the ergot alkaloids getting into the seed, into the flour? And we've identified 2 routes, one a physical transfer, direct contamination, but also the alkaloids appear to be able to move in planter. So what is ergot? Ergot is a fungal disease. It's a fungal disease that infects the flowers of cereals and grasses. In cereals and grasses, it's caused by Claviceps purpurea. The spore gets into the flower and fills the cavity where the seed will develop with a fungal mass. That fungal mass, after 14 days after infection, produces the asexual stage, which is honeydew. This is a sugary solution produced by the plant, and we assume that the fungus can manipulate the plant to produce this sugary solution, and the fungal spores are suspended in that honeydew, and that provides a source of infection which is then transferred to other flowers on the ear or to other ears. Then about 21 to 28 days after initial infection, you get these sclerotia, these dark ergot sclerotia that are formed where the seed would normally develop, and these are the overwintering structures of the pathogen. They fall to the ground, they lay on the soil, and come the spring, they germinate. So why are we concerned about ergot? Ergot doesn't really impact yield. It doesn't have a big effect on the amount of seed that are produced by the crop overall. But what it does to have is loads of alkaloids, extremely toxic alkaloids, as Joe has demonstrated, that cause some very nasty symptoms. And these alkaloids are thought, again, no strong evidence, to protect the ergots, to protect the sclerotia as they lie on the soil from predation by insects and other pigeons or pests. So they don't get eaten, they just lay there. So what do we know about the infection cycle? So this is an ergot. Early spring, April, May, it germinates, it produces the asexual cycle, which produces this periphecium. Within this periphecium are the conidia, which then pass, the conidia here, is deposited onto the stigma. So this is a wheat ovary. This is what would develop into a seed. It lands on the stigma, and it grows down that stigma through the style down to the base of the ovary. And we have some evidence to suggest that what the pathogen is doing, it's fooling the plant into thinking it's a pollen grain, because we've got no evidence that the plant mounts a resistance response to this spore germinating on the stigma and growing down to the ovary. Yeah? We've got evidence looking at gene expression, hormonal changes, that says it's fooling the plant to think, oh, I'm being pollinated. And we also know that if you pollinate, if pollen actually gets onto the stigma before the pathogen spore, then the pathogen can't infect. Okay? There's a balance. So after 2 to 5 days, you get this mass, this green here is the fungal tissue, which grows and fills up the ovary cavity. And we know that within 7 days, you get this, what's known as a facetial product, which basically is just a mass. You can see that's the ovary, that's where a seed would have been, but that's actually infected with the fungus, and you have this fungal mass. 14 days, you get honeydew produced from that infected floret, which is full of additional conidia, which can go on infect other florets. And then, as I said, 21 days, 28 days, that fungal mass produces a hard coat and forms a sclerotia. And this is a very hard, resistant body. Believe me, I've tried grinding them up, and they are tough. Yeah? So between 2005 and 2009, before I joined NIAB, colleagues of mine undertook a project to try and look at some of the questions of how the epidemiology of this pathogen on the farm. And one of the things that was bandied about, that where is this pathogen coming from? What is the source, the primary source of infection? And there's a lot of comments that wild grasses on the farm, in grass margins, weeds within the crop, of acting as a source of infection, primary infection for the crop. So I undertook a study of a lot of grass margins on farm, and it discovered that ergots were found in 37 grass species. Okay? But most commonly in crouch grass, Italian ryegrass, black grass, perennial ryegrass, cocksfoot, etc., etc. But what they did discover is that if you took the ergots off those grasses, you could take that isolate, that single isolate, produce conidia, and reinfect wheat with that conidia. And the ergots that were found on black grass, couch grass, Italian ryegrass, and perennial ryegrass were particularly good at infecting wheat. Okay, so there's this cross-species infection. Okay, and in fact, a lot of the studies we've done here at NIAB, we actually use as our main isolate, which is most aggressive, originated from black grass. Okay. So our current thinking is that we have a life cycle for this pathogen in the field, which consists of the sclerotia germinating around April, May, and those initial conidia probably affect wild grasses. Okay? The wild grasses then produce honeydew, which are full of conidia, these sticky, sugary solutions, which are transferred either by rain splash or by insects. And again, another one of these open questions. We don't know which is the predominant method of transfer, But those conidia contained within that honeydew, which you can imagine would attract insects, is then transferred onto the crop as a secondary source of infection. Okay? Now, this is some work, so as Joe mentioned, that France has a particular problem with ergot as well. So Virulence in France has done a number of studies over the past 10 years, looking at ways of coming up with recommendations of how you control ergot on farm. So I've tried to summarise their ergot management tool, which they put up on their website in 2021. It's in French, so it was a bit of a struggle. I had to check my French translation on some of these. But basically, first of all, they say, right, what's the risk that you could have ergot in your farm, in your field, on your farm? So they ask the farmer, have you seen ergot in the past 2 years? No or yes. Then they say, to what depth have you tilled? Okay? Because if you deep till, you bury the ergots. And if you bury them deep enough, when they germinate, the spores don't come to the surface, or they can't germinate because you've buried them. Okay? But if you shallow till, along with recommendations of DEFRA policy for regenerative farming practices, those ergots can germinate and the conidia reach the surface. What we also know is that seed merchants can sell certified seed which contains ergots. Okay? Or if you've kept farm saved seed and you haven't got rid of all your ergots, they say, have you got ergots in the seed which you're about to sow? Okay? So based on those questions, you have a low, medium to high risk of having the initial inoculum on your field or ready to go into your crop. So the next question you then ask yourself is, what is the risk of that ergot establishing an infection in my field? Okay? So you know whether you've got a low, medium, or high risk of the inoculum, of the ergots being present. Do I have good weed control? Because we can see that this pathogen can move quite freely between common grass weeds that we find in our cereal crops, like blackgrass, okay, onto your crop. So if you haven't got good weed control, yeah, you're at a higher risk of infection being spread around within that ecosystem. Then the French advise, do you have weather conditions? I'm not sure that it ever gets to below 4 degrees, even in France, during the spring and summer, but that's what's on their website, and I did double check. So if the temperatures get low, or you have more than 44 millimetres of rain, you have a higher risk of that infection establishing within your cereal crop. Okay? So again, that is your risk factor. So what are their recommendations? What can we do at present to try and control that? So primarily, you need an effective control of your weeds. Yeah? And I think a lot of people in this audience will know that that is another big issue facing the industry, controlling weeds, especially breeds like ryegrass and blackgrass. Okay? You can control it by deep tillage. So if you have ergots, a risk of ergots in your field, you can do a deep tillage in 1 year to bury those ergots. And then the following year, do a shallow tillage so that you don't bring those ergots back to the surface. Because again, Virulence have data that say that these ergots, these sclerotia, these overwintering structures, can survive from anything from 1 even up to 5 years. They can still be viable. Okay? Avoid growing susceptible cereals for at least 2 years. Okay, so your rotation, your crop rotation. And if you do have ergots in your field, harvest those areas separately. Okay, so you're not putting them into the bulk of your harvest. Okay, so that's the current recommendations that Virulence and that came out of the LINK project that we did here. So, ergot resistance. Well, we don't breed for ergot in the UK, primarily because it's not a disease that affects yield. It does have particularly more implications for rye. So where, as you know, in parts of Northern Europe, rye is a bigger crop than it is here. Rye is an open flowering cereal, as opposed to wheat and barley, which are closed flowering. And because of that open flowering nature, where the flower droops, yeah, and it's cross-pollinated, there's a greater chance of the pathogen getting into those flowers. So ergot in rye is a really big problem. And in fact, the limits for ergot's alkaloids in rye, as Joe will tell you, are actually, the EU accepts that they will be higher because of that. But we have looked at resistance, and at NIAB we have developed systems for measuring this disease, for assessing the level of disease. We assess honeydew, the amount of honeydew that's produced, based on a 1 to 4 scale. But we also assess the sclerotia, the size and the weight of sclerotia, and we can use a Marvin seed analyzer to analyze the sclerotia. And you can see here we have a scale that assesses the sclerotia. Using those phenotypes, we've assessed disease resistance in a number of wheat varieties, and the work, initially we screened a whole host of wheat varieties in the field. We decided that Robigus and Solstice represented a resistant and susceptible variety. However, when we crossed them and made a mapping population and looked at the genes that conferred a reduction in sclerotia size and weight, we found 4 QTLs. 2 of them came from Robigus and 2 came from Solstice. What we also found is that the QTLs on 4B and 4D co-located with the semi-dwarfing alleles. For those who don't know, in wheat, there is a gene that affects height of wheat. And the breeders have introduced into wheat alleles at these genes, which make wheat, rather than being 6 foot tall, as it was in the 50s, to being down here. Semi-dwarfing genes, okay? So we found that some of these QTLs which reduce sclerotia were co-locating with these semi-dwarfing alleles. So we tested this further by looking at near isogenic lines in an old variety called Mercia, into which different alleles at these RHT genes have been introduced. So we have alleles 1B and B1B, B1D, B1C, B1C. Okay? So 4 different alleles at 2 different loci. Okay? And the C allele actually makes the wheat very small. So it's a really hard dwarfing allele. And yes, we found that if you have those genes in the wheat background, you do reduce the size and the weight of sclerotia. So fortuitously, in breeding for semi-dwarf wheats, we have bred for a small amount of resistance. But again, it's only partial, it's not complete. And that's what we found with all the resistances that we've looked at for ergot. There's no gene-for-gene reaction, there's no major resistance, this is all partial resistance. Okay? And this also helps us to get to a theory that, so these genes, these RHT genes, produce a protein called DELA. And in plants, gibberellins bind to DELA's proteins, degrade the protein, and when that protein is degraded and removed from the system, the wheat plant grows tall. In the semi-dwarfing wheats, the DELA protein is altered. The gibberellins can no longer bind and degrade the protein, so the plant remains semi-dwarfed. So again, we've got a very complex system in the plant, okay? And DELA proteins have actually been shown to be involved in a whole host of other traits within wheat that are playing into this pathogen's normal infection process. Okay? And this may come back to the theory that I presented earlier, that we believe the pathogen is trying to fool the plant into thinking it's a pollen grain. Because one of the things we see that in those, the stigma and the style and the ovary, when you add those, you put the pathogen conidia onto that stigma, you get the gibberellic acids, gibberellins, go up. Yeah, the profiles change completely. And we also see changes in expression of these RHT genes. And we're working with the team at Rothamsted, who are experts in this area, to try and investigate this area a little bit further. The other source of resistance we found was in a durum wheat called Greenshank, and this is part of a collaboration we've had for some years with a Canadian group in Morden. And there we found a source of resistance on chromosome 2A, which actually reduced the level of honeydew. And we'd not seen that before. So here you have a resistance that could reduce the source of infection, that secondary infection, yeah? That honeydew that gets moved by insects from an infected flower to an uninfected flower. So again, that's a possible source of resistance that would be benefit further study. Okay, so alkaloids. There are 6 main ergot alkaloids that are produced by this pathogen, and they have and they're emitter forms. And I'm no biochemist, so don't ask me about details about these alkaloids. But we have shown that the genes in the pathogen that produce these alkaloids, the biosynthesis genes, are expressed in the infected ovary. There you go. By 5 days after infection. So they're expressed very early in that infection process. I just want to skip on one. So if you look at the different pathogen tissues, so if we take the honeydew, which is that sugary solution containing the pathogen spores, if we take the Ferrocecea, which are produced 5 to 7 days after infection, that fungal mass formed where the seed would normally form, and we take the Sclerotia and we grind them up and we measure for alkaloids. And this is measurements that were done by Camden BOR, where they have a certified system, accredited system for measuring alkaloids. You can see that in honeydew, well, you can't even see it. It's not even registering on this scale. So for Celia, you start to see levels of ergot alkaloids produced. But the most of the alkaloids are in the Sclerotia. Okay? So we know the honeydew is not causing problems in delivering alkaloids to clean seed, because Joe mentioned that we have evidence that clean seed that's developing in that ear can contain alkaloids. So it's not coming from the honeydew, so where is it coming from? Can I go back? Okay, so we had a project funded by AHDB where we posed 2 questions. We wanted to determine how the alkaloids are being transferred from flowers infected with the pathogen, with claviceps, onto healthy grain that was developing within that same ear. We also wanted to determine whether the alkaloids that are seen within Sclerotia can be transferred physically to grain during harvest and transportation to the mill. Okay, so we wanted to compare those 2 potential mechanisms by which the alkaloids could find their way onto clean grain. So we developed an inoculation system, and here we would inoculate the middle florets, so hand inoculate individual flowers with Canidia of the pathogen. We'd then protect the remaining part of the ear from any cross-contamination. Okay? We would then wait until we would let the ear develop, and we'd collect the seed. We'd collect the seed from the top part, and we'd collect the seed that developed beneath. Okay? And for this, we used our favourite isolate, this one, which came from blackgrass. And I think this, that's in a single variety. So what we found, the data is variable. We did these studies in barley, in rye, and in wheat. We used, as I said, a single isolate, and we took the grain that developed above the infected florets and the grain that developed below the infected florets. And we found massive levels of ergot alkaloids in that grain. Levels that varied in wheat from 0.2 to 160 milligrams, when the EU regulations are 0.1 milligram. Okay? Again, barley, not quite such a big range, and rye, not a big range, but we found alkaloids on those otherwise clean grains that would have gone to mill and turned into flour. Okay? So yes, we know that the ergot alkaloids can move in planter from an infected floret onto healthy grain developing within that year. We believe it's probably an active mechanism involving plant transporters, and we've established collaborations with various groups in the UK to address that question. Because we also want to know if it's transferring in planter, where in the seed is it? Is it in the endosperm? Is it in the embryo? Is it in the seed coat? There's lots of questions that we want to answer. The second part of this study was to look at physical transfer. Can these alkaloids be physically transferred from a sclerotia, either whole or broken, onto grain? And for this we did tumbling experiments where we basically mixed seed, clean seed, with broken or whole sclerotia. And this work was done by Camden BRI, who also do the alkaloid testing. And yes, we did. We found that control, no alkaloids. If you mix wheat seed with obviously broken sclerotia, you get more alkaloids transferred onto the seed than if you use whole sclerotia. If you use a great quantity of sclerotia, you get more alkaloids. But these levels that we found are exceedingly lower than what we found with the implanter transfer, which is worrying. Because even if we get a load of grain and we clean out the sclerotia, we're not ruling out the fact that that grain is already contaminated, and we just haven't seen it because it's been contaminated on the plant while it's growing in the field. So, take-home message. If we're going to get a control on top of ergot alkaloids, we've got to do it on farm. Full stop.
Okay. We've got Sasha. I'm not introducing you again.
That's all right. Yeah, it's only a really brief talk for me. You might be relieved to hear. So this is just to talk to announce that the AHDB have commissioned a new piece of research about ergot. So we know we're seeing more grass weeds in the rotation. Field margins are becoming a bigger part of our landscape, and you know there's a move to less tillage as well. All of these are risk factors, and on the back of that, we're seeing bigger issues with ergot in crops. So last summer, AHDB published a call for proposals looking at updating the guidance on ergot management. And in the autumn, that was awarded to ADAS. So that's a team that's been led by Philip Bounds, Anastasia Sokolidi, Jonathan Blake and Sarah Cook, and supported by various other pathologists at ADAS. So a very good and capable project team. So they're going to be focusing on carrying out literature researches, so scientific literature, grey literature, the sort of essentially AHDB style sort of publications, to look at various things. So what are the key life stages of ergot, and where can we use those life stages to sort of improve our management. It's going to look at the current regulations, the implications of those. It's going to identify crop management practices that growers can use now to help them manage the disease. Where there is ergot in the crop, what are the harvesting techniques that can be used? Where there's ergot in the grain, what are the sampling practices that are best for that, and how they can be removed most effectively. It's going to explore opportunities for plant breeding, and then it's going to come up with conclusions and recommendations. So there's going to be three main outputs from that piece of work. So the AHDB ergot management guidelines are going to be updated. They're going to focus on practical recommendations for management of the disease. So pre-planting management, within crop management, and then post-harvest management. And then it's also going to be identifying knowledge gaps. So where we can prioritise future research funding for the management of this disease. That's going to be available, I believe, in the spring, so hopefully in time for some in-field management to occur this season, but certainly in time for next season, pre-planting management guidance to be considered by agronomists and the growers. And then finally, there's going to be an ergot identification guide produced, and that's going to be available in the autumn, and that's going to be helpful for the users to identify where they've got ergot in their crop, or is it in grass weeds within the crop, or in the margins as well. And yeah, so that'll be available later on this year. So yeah, watch this space, and there's going to be more information being disseminated by ADAS and AHDB later on this year.